As we know, proteins are an important part of diet. They provide amino acids, the building blocks of, well, other proteins that we use in our body for various tasks, such as structural. Collagen is the most abundant protein in animal bodies. It is considered one of the hallmark characteristics of metazoans, so animals. Well, true animals, so to speak. We don't usually quite include, say, sponges, which are considered simple animals. Now, nucleic acids, well, we're also familiar with those if we've been talking about DNA, deoxyribonucleic acid, and RNA, so ribonucleic acid. Now, amino acids, well, they come in many different types, but they share a few common characteristics. So, humans are considered level amino. What this actually means is the fact that, hey, these four groups are not the same. Whoa, no two of them are the same. What does that mean? That means in space, like say, oh, suppose H is up here, and then the amine group, which usually exists in charged form in relatively neutral solutions, but can be idealized as this. And then we have COOH, and out here we have the R group. Now, except for glycine, which has just a hydrogen for the R group, what happens when we put this through a mirror? They're not the same molecules. They do not behave the same way when it comes to interacting with anything else that is stereospecific. So, where the orientations matter. Now, I'm pretty sure we all know that orientations in real life do matter in the form of, hey, if you're right-handed, writing with your left hand is going to be a bit of a problem. Yeah? Okay. So what does level amino actually mean? Well, this has to do with what happens when we take a solution of these amino acids and we shine a light through it. Which way does the light end up bending? Now, this is not actually a crucial knowledge at this point in our study of biology. It's just something that we should be conscious of, that the orientation of groups in space is very important. This actually goes a lot into, say, enzyme function, and is why dextroamino acids are not so convenient for our digestion. Okay, so what does having both an amino and a carboxyl group do to a molecule? Well, when you put it in neutral solutions, it tends to exist in this form. This is called a Zwitter ion. Or you can say that this is the Zwitter ionic form. So here, the amino group, well, we know that, okay, in ammonia, in a solution of ammonia, most of it remains uncharged. So why is it that here there's this proton binding? Guys, if you buffer this solution to near neutral, then you will find that of those ammonia molecules, a lot of them will be charged. Even though, if you just have ammonia in a solution and you don't buffer it, so just let the pH be whatever that concentration of ammonia gives you, most of the ammonia will be uncharged. So what about the carboxyl? Well, when you have, say, vinegar, not very much of the acetic acid is actually ionized. Most of the hydrogens stay on the acid. But what happens when you start making the pH bigger? So making it more neutral. Then you'll have more and more of the acid dumping the protons into the solution, where they are probably promptly locked up by whatever buffer you're adding. So what happens when you have a solution of an amino acid? Well, yes, okay, the pH isn't going to be perfectly neutral. They don't have perfectly corresponding pKa's, or rather binding coefficients for protons. But you will generally end up with a solution that is mostly in zwitterionic form. So positively charged on one end, negatively charged on the other end. Now, an amino group and a carboxyl group can bind to each other in the form of an amide bond. So they end up locking each other up. Besides forming the amide bond and also forming the opportunity for some hydrogen bonding right there, which is responsible for, say, the alpha helix or beta pleated sheet for that matter, the main determinant of an amino acid's behavior is the side chain, the R group. So we can have an R group as simple as a single hydrogen atom, so for glycine, or we can have alanine, or valine, leucine, isoleucine. Hey, wait a second. Do we notice a trend here? What about a hydroxyl or a carboxylic acid as another side group? These here are all non-polar amino acids. They are sometimes referred to as aliphatic and aromatic. Aromatic means that you have a benzene ring or a structure that's derivable from a benzene ring. Now, that actually overlaps a little bit with a polar amino acid. See here, there's a benzene ring here. Benzene rings have special chemical properties, which we aren't going to go over yet. Now you probably noticed that one of these amino acids is not like the others. No, I'm not talking about glycine, even though glycine also does some interesting things to the structure of, say, an alpha helix or a beta sheet. What about this, proline? Proline has very different characteristics from these other ones due to the way it bonds to things. Now, what does it do when you take an amide bond and lock it at a different angle from all the other amide bonds in, say, a zigzag pattern? You break the pattern, of course. And, well, for uncharged polar amino acids, what can these do? Hey, we can do some hydrogen bonding, potentially. Or what else could we do? We could, due to the structure, have all the nonpolar ones pointed inward, or in the case of cysteine, participating in disulfide bridges, whereas the polar and charged amino acids point outward. Now, you will find a lot of these in the active sites of various enzymes, presuming whatever you're dealing with is either charged or polar. If whatever molecule you're dealing with is not charged or polar, well, distributions may vary. There's another reason that a lot of them are found in the active site. Guys, the active site of enzymes is on the surface. Energetically speaking, it is very favorable to have these hydrophilic groups sticking out into the surrounding aqueous solution because cells are mostly aqueous solution. There are some exceptions though. Sometimes you will find nonpolar amino acids on the outside. When? When is this possible? Hey guys, what if we have a protein that sticks through a membrane? Well, we know that the membrane is kind of like this. Now the heads are hydrophilic. They point outward into the aqueous solution, whether that be inside or outside the cell. Great. Okay, what about the tails? Hey, they're hydrophobic. So you're going to want these aliphatic, or aromatic. But regardless, non-polar amino acids sticking out in this area, while you have your hydrophilic amino acids elsewhere. So non-polar, we might as well just label this hydrophobic. Now we do point out though that mileage will vary on how polar these are and thus how hydrophilic they are. For example, tyrosine is not quite as hydrophilic as just calling it an uncharged polar like serine would have you believe. Just because the benzene ring is huge and not exactly hydrophilic. Now, proteins, of course, we know that proteins can be used for all sorts of things. So structural proteins, hair, tendons, and ligaments. Hair is mostly keratin. Tendons and ligaments are mostly various collagens. These function for support and elasticity. But there are plenty of non-animal examples. For example, all eukaryotes have cytoskeletons. What are they made of? Well, microtubules, an essential part of cytoskeletons, hey, those are made out of tubulin, which is a protein. Now, what other proteins can we do? Well, we can use proteins for defense, such as antibodies or immunoglobins. So they function to fight infections. Well, depending on our idea of defense, we could also say that various other proteins function in defense, such as, what is an essential part of not bleeding? When, say, we get a paper cut. Well, part of that is maybe the paper cut's just not deep enough. But if it actually breached a capillary, hey, we have fibrin from fibrinogen. And that's a protein, and it functions in defense. And for messenger proteins, well, hormones, some of them are polypeptides, or just peptide chains, and we just lump them in under proteins. Because really, a protein can be made out of several polypeptides, or one. Which means, even though for now we generally call over 50 peptides linked together a polypeptide, really they're all still proteins. So as we get further and further into biology, these distinctions that we start working with, we generally just hand wave them aside. Now, we could use proteins to transport things, such as hemoglobin, what makes blood red in humans and mammals, and a lot of other creatures. But we do point out that there are other globins that also carry oxygen, and they don't use iron, in which case we might get blue-colored blood or greenish blood, as we find in, say, certain mollusks. Now, proteins can also be used as recognitions and receptors, so say major histocompatibility complex and various other receptors that we find, such as, well, the receptors we use for scent are proteins. Well, we can also use proteins as catalysts, as in the case of amylase, the enzyme that we use to digest starch. So generally we talk about salivary amylase, which is found in saliva, and is the reason why if you put a soda cracker in your mouth, sure, it starts off tasting salty if it's been salted, but very quickly it starts tasting sweet. That's because the amylase in your saliva is breaking down the starch into sugars. Now we can also use proteins to move around, such as actin and myosin, which are essential components of muscle and also of intracellular, so inside cell transport. What else could we use? Guys, what about flagellin? So the protein that is used in bacteria for their flagella, which are very different structurally from eukaryote flagella. Well, that's a protein, and it's used for movement. Okay, so what are proteins? Well, they're made of amino acids. How? Well, we form what we call a peptide bond. This is an amide bond formed between an amine group and a carboxylic acid. Now, a polypeptide has more than 50 amino acids, but you'll notice that in later grades this distinction basically just goes away. We just call all of these things polypeptides. Because after all, a peptide bond is this thing. And if you have more than 2 amino acids, so say 3 or more, what do we have? We have multiple peptides. Just like polygons are many-sided shapes, we just go with polypeptide later. Okay, so this is a condensation reaction or a dehydration reaction. So how can we break these apart? Well, hydrolysis is what breaks up proteins into their separate amino acids, whether inside our own cells, because some proteins get old and damaged and worn out, or just are no longer needed, and thus they get broken down for parts to make other proteins with, or to be metabolized. Humans don't have much of an amino acid reserve, except counting our musculature. We generally metabolize the proteins that we take in, that we don't use to build new proteins right away, and we store the energy as fats. This is why eating a protein-rich diet, well, it kind of puts a load on your excretory system because you have to get rid of the nitrogenous waste in the form of urea, which is found in urine. And it gains weight. And what else? Well, wait a second. It could help you build muscle if you actually expend enough energy and you prompt your muscles to undergo self-repair and enlargement. Yeah, but a lot of people who eat high-protein diets don't do this. So all they're doing by eating proteins too much is getting fat, pretty much, and loading down their excretory system. Now, how is it that we go from these amino acids to something that can be joined together to make fatty acids? Hey, wait a second. In fatty acids, we have no amines. Deamination is required. That's why we have to secrete the extra nitrogenous waste. So, essentially what I'm saying is eating too much proteins under the idea that it is healthy to do so, healthier than eating fatty foods, for example, no, not really. Generally, human energy intake should be large majority carbohydrates for a healthy diet. Okay, we notice that with a polypeptide, we will have one end showing an amino end, and the other end is a carboxyl. So we have an N-terminus and a C-terminus. Now, how are proteins synthesized? Well, given the fact that English is a left-to-right language, and this diagram has N on the left end and C on the right end, we point out that proteins, when we list the amino acids, we list them N to C. So synthesis is also N to C. We add new amino acids at the carboxyl end. So here we notice this being illustrated another way. Why? Well, it's because we want to illustrate these possible structures. So protein structures, well, they can be broken down. And initially, when a protein is first synthesized, it does not have structure. It's just a long chain of amino acids. What happens to give it structure? Well, it randomly flails about, pretty much, until you get hydrogen bonding in particular patterns that are partially dictated by what side chains you're dealing with. and partially corralled into it by chaperonins, so chaperone proteins that help these proteins fold correctly. But the point is that, hey, if we get this structure out of this blue sequence, which honestly should be read from N to C, and this pink structure here, well, it just so happens that our diagram is more convenient with N on this end. But don't forget that when we list these things, we go from N to C. And that when we synthesize proteins, we go from N to C. So N is the initial amino acid. Okay. You might have heard of the term amino acid residue. Why residue? Well, when we synthesize these things, we lose certain parts of the amino acid because it's a dehydration reaction. And what does that do? Well, what's left? The residue, right? When you take part of something away, say dry something up in, say, a petri dish or whatever, you end up with residue. Okay, so we have the primary structure, which is the sequence of amino acids, and then that will spontaneously fold into secondary structures. Sometimes this requires a little bit of help, though. And the secondary structures are potentially alpha helices or beta pleated sheets. Now, beta pleated sheets can be parallel or they can be anti-parallel. What does this mean? Well, okay, we could have, this, and then, so point that way versus point this way. So that's anti-parallel. On the other hand, it could be the case that this just bumps up against another section of the protein, which also forms the same zigzag structure, and hey, they end up being parallel. Of course, then you have to have the connector parts in between. So that's a big difference, yeah? And alpha helices, are they left-handed or right-handed? Well, both types of structures exist.